So I'm going to talk today about possibility, the early stage possibility of providing to you guys a handheld device, an ultrasound device that can, I think, detect midline shift in traumatic brain injury patients. Pardon me. It's funding from the DoD, and they have a particular problem. They have soldiers who are often far away from care, and they experience some kind of head injury. And they need to know what to do with these patients. They can't just call up the ambulance. It's not a trivial exercise to call up an ambulance and have people picked up because the ambulance might get shot down, for example. So people on the ground are faced with the following problem. They want to know if this individual who's perhaps lying there semi-conscious or unconscious, is it worth, air quotes, triaging them and getting them care? Life has intrinsic value, so in principle, yes, but there, as Dr. Ellenbogen and others know, it's a non-trivial decision to decide to medevac someone in far forward circumstances. So what they really want to know is if they medevac someone, is there a chance they can improve that person's clinical outcome? Because if not, they'll make some other decisions based upon the circumstances on the ground. And so at the end of the day, they want to know, is there a neurologically treatable, typically neurosurgically treatable lesion in a person's head? And you guys know much better than I all the different versions of treatable or untreatable lesions in the head. If it's an epidural, evacuation, craniectomy, for example, will do really good. If it's an interparenchymal bleed, well, outcomes are typically not so good, and maybe it's not worthwhile with all the other concerns to evacuate this person. Maybe someone else there's a better chance of survival will get medevaced first. So we developed a device with a company called TerraSon outside of Boston. It's a tablet-based device. It's the size of your iPad in projection, double the thickness though, and of course has a keyboard and a probe. And otherwise it's diagnostic ultrasound, but we analyze it in a novel way. It turns out that the brain pulsates when blood enters the brain and then leaves the brain. So on your left is a so-called B-mode image of the brain of a healthy test subject. It's an axial image. The bright arc along the bottom of that pizza pie slice is the skull. Ultrasound comes in from the top, strikes the skull, backscatters, and you get this bright area. You might be able to see the midline as the horizontal line, white line going through the middle. And if you grab a pixel in the close to the probe relative to the midline, then which is the blue, you'll see the brain displacing on the order of 50 microns. And if you grab a pixel on the far side of the midline, you also see brain pulsation on the order of 50 microns, either away or towards the transducer, which I denote on the slide. So just to give you context, if you look at the white part of the nail on your fingertip, that's typically a millimeter across, pardon me, the hair on someone's head who has hair, that's typically has a diameter of 100 to 150 microns. So this is really small displacements that ultrasound can measure. So Department of Defense funded us to collect a bunch of data and analyze it. And here's an example of the data. If you look in the upper left, that's an ultrasound plane. Ultrasound images are typically in a plane. Pardon me. If you look in the lower right, that's a GIF of the skull of a patient with a craniectomy with the ultrasound plane projected into it. And the intervening images show either the CT plane alone that projects onto that ultrasound plane. That's the upper right, for example. And then we in the intervening images C, D, and E, we highlight one and make the other one go away so you could see how the ultrasound image projects onto the CT plane. A whole lot of work went into generating about 130 data sets that have this property. While trying to develop images based upon the pulsation of brain, we noticed that if you average in time and space that pulsation a great deal, in this case for a healthy test subject, the symmetry of the pulsation in each hemisphere really stands out. So on the left is a beam mode image, again, of a healthy test subject, the same one as before. On the right is that average brain tissue displacement image. So the near field hemisphere displaces away from the transducer, that's the blue. The red displaces towards the transducer. We looked at this image and many others like it, and we said, we wonder if it's possible to detect midline shift. I'm sorry, I'm a little coffee this morning. So we went about attempting through a variety of modeling efforts whose details I'll skip to detect midline shift in our traumatic brain injury patients and in healthy test subjects as a negative control. So the hypothesis, pardon me, supported by some MRE imaging that I will show you in a minute, is that the brain has a center of pulsation. Cerebral spinal fluid moves in and out of brain, blood moves in and out of brain in a way that the brain tends to lift up and out and then down and in, which we can detect with ultrasound, and others have detected and quantified rather with a magnetic resonance elastography. I need to go back to Iceland, clearly. So we discovered that we could detect a plausible center for pulsation using brain tissue pulsatility developed in a map. And it was motivated by that previous movie that I showed you and a recent paper on magnetic resonance elastography. So I'll show you a couple movies from that MRE paper. So healthy test subject again. If you look in this coronal section, you could see the brain going up and going down and expanding in and out, mostly up and down. So that's one example. Really cool paper, I must admit. So here they've highlighted the cerebral spinal fluid, and you could see all the ventricles there, and you could see them pulsating. Again, from this image set of movies and our own work with ultrasound, we posited that there exists a center of pulsation. Here's a still image that puts vectors on that displacement in sagittal, axial, and coronal images. And then finally, here's another movie that, again, motivates this idea. There's a center of pulsation in healthy brain, which should sit in the center of the cranium itself. And with lesions in the brain, most notably bleeds of various sorts, it seemed plausible that that center of pulsation would displace from the center of the brain. So again, for our healthy test subjects and for traumatized TBI patients, we have multiple images that project onto a CT image through a lot of work. That's on the left. On the right, this is a GIF showing many such images with the center of pulsation derived, highlighted rather in the middle in the hot color. So again, lots of math here. You could all do it, but I don't want to take up your time discussing it. At the end of the day, in each ultrasound plane, we create a mathematical model of in 3 dimensions of what that center, what the brain would do if there's a center for pulsation that's maximum in the middle and decays with a Gaussian distribution going out to the edge of the skull where it doesn't displace at all because the skull holds back the brain. And we projected that model onto each of these ultrasound planes, anywhere from one on either side of the brain for some patients where there was difficulty collecting data to say 15 or so images on either side of the brain. And in each plane, we then calculated the center for pulsation and then collected them together, and through some more regression analysis, determined where that center lies in 3D space. So somewhere in the cranium is something we call the center for pulsation. And we project that onto a plane, which would be a plane that defines the midline of the brain of a given patient or healthy test subject. Now, we had to do some work to clean things up. For working with your patients, your TBI patients, Anne Moore and her wonderful crew, the cerebral vascular ultrasound people, held the transducer by hand. And so even though they're incredibly steady, despite the coffee that they drink, we had to get an estimate of how the transducer held against the head of a patient moved over, say, a 8 or 10 second period. So in the lower left is skull displacement over the time we collected a given set of images for a traumatic brain injury patient. And in red is the axial displacement. So it's the displacement towards and away from the patient's head. And amazing, it just hardly moves. This is, again, held freehand, but tended to move up to a millimeter in the lateral direction. So in other words, Anne Moore and her crew, that has some slight twist to the transducer as it was held to a patient, but incredibly steady. So we got an estimate for the velocity in the brain unfiltered without taking this out. And for example, at about 6 seconds, the middle set of images shows you brain tissue displacement. That's the upper image, gold versus blue, towards or away from the transducer, where you can't really see much structure. And below that is the unfiltered versus the mathematically filtered displacement caused by movement of the transducer. So we really do clean it up nicely. And then in the far right is, well, what does the displacement look like inside the skull once we do this filtering? And again, thanks to the mathematical modeling of colleagues of mine, the displacement is kept very small. And out of this analysis comes the 2 hemispheres of brain that we saw back for a healthy test subject where everything was perfect. He was still, there was a headset, etc. A whole lot of work went into that. So we could detect the 2 hemispheres and then proceed to project the mathematical center for pulsation into all the different ultrasound images we had, themselves projected into the CT image. For our healthy test subjects, we're able to develop a very kludgy but functional headset, put patients on a massage table, healthy test subjects on a massage table. We have 2 of these handheld, 2 of these diagnostic ultrasound devices. And we put it on the heads of healthy test subjects to get a sense of when you, essentially negative control. These people, they're all in their 20s, there's students of mine and their friends, and the question was, when everything is perfect, how well can you determine the center of pulsation, which should be in the center of these patients, these healthy test subjects' heads? We also developed an algorithm that projects the center for pulsation in real time. We have 2 of these devices. You could use them tomorrow with IRB approval, or if you guys just decide you want to put it on a patient. And for our healthy test subjects, we collected ultrasound from a whole variety of angles to really explore what an optimal angle is, where everything is held steady, etc. And these are, if you look at the red plus sign, that's where the predicted center of pulsation is for these healthy test subjects. Again, these are B-mode images based on backscatter. The skull shows up as this far side skull shows up an arc along the bottom of this pizza pie. And I think you could maybe see it a little bit better on the right-hand side. The B-mode images show just about nothing. And as it turns out, this plus sign is rather stable. Depending upon the angle, they're within approximately a millimeter of each other where the center of pulsation is. And I know apply to healthy test subjects and to traumatic brain injury patients. Once you hold this thing steady, we get actually a really good estimate of where the center of pulsation is. So let me give you some numbers now. So for our 10 healthy test subjects, we see a range of values is averaged over, in this case, 15 images from either side of this healthy test subject. It takes about 15 minutes to collect this data. And on average, the center for pulsation is within a millimeter plus or minus half a millimeter of the center of the skull. And for all these people, all these 20-something people, we could see the midline in the B-mode image rather nicely, and that was our gold standard. So good news for a healthy test subject for whom there should not be any midline shift. There's some uncertainty in our measurements, about a millimeter, which is well within the plus or minus 5 millimeters that tends to get the neuroradiologists and neurosurgeons excited and can motivate a neurosurgical procedure. So I'm almost done, Rich. Here are some results that'll take me a couple minutes to describe. Up above is a comparison of midline shift as detected by our radiology colleagues. And along the vertical axis is the center of pulsation derived from our algorithm. And you'll see two regression lines there. If you look in the upper left, there's a purple one where we use all the data, including that from a single patient who had a very large midline shift as determined by the radiologist. And then in green is the regression line with where you take that patient's data out. So not great. And here we took all comers, basically all the patients we had where there was at least one ultrasound image whose quality was sufficient to do the analysis on either side of the brain. Now, if you look down to the lower two images, on the right is a Bland-Altman plot, on the left is the same kind of image. Radiologists determined midline shift along the horizontal axis, center of pulsation along the vertical axis, and now we required at least 2 scans, often more, of sufficient quality in image quality and in registration. And there the regression is significantly better. So recall that we had access to these patients after they got to the ER, after they were triaged, and we only got the ones where they did not receive a neurosurgical intervention with a very small handful of exceptions. So this isn't bad. It's not a home run, but for the most part, we get a prediction of midline shift based upon the center of pulsation that might have some diagnostic utility. What I'd like to do, and at least Rob Bonner and I hope to write a grant to support this, is get access to patients as they enter the ER and before a neurosurgical intervention to get some patients with a large midline shift. So we're able to collect this data in real time. We just need a small handful of images on either side. We can do it freehand. We don't need a special headset. And I have hopes that with another grant, which again, I hope to write with Rob Bono and with Terrason, this ultrasound company, that we could provide to you guys a device that can predict the center of pulsation as a proxy for midline shift, useful in an ambulance, useful in far forward circumstances, all potentially. And that could quickly give you an estimate of that midline shift. As I'll close by noting that we collected data from some stroke patients, and I had hoped to show you some examples, and I couldn't find them for this morning's talk. So there, I just have to describe it to you, and then I'll close. For these stroke patients, radiologically, they don't have a midline shift, but they have significant differences in perfusion in one hemisphere versus the other. So I'm going to go way back to this movie, and I'll close with this movie and then acknowledgements. So these are patients where their center of pulsation is significantly shifted away from the midline, even though there isn't a midline shift. And I'll note that we've hypothesized that the center of pulsation is a useful quantity as a proxy for midline shift for people who have a meaningful lesion in their head. For the stroke patients, what they have is significant differences in cerebral perfusion, one hemisphere versus the other. So I think this algorithm, this algorithm, in our view, is clearly a measure of cerebral perfusion, left versus right. And for ischemic stroke patients, for example, there's a significant difference in perfusion if you have a blood clot, for example. And so this device might have utility in time in helping triage stroke patients as well. And I have Mike Levitt to thank for allowing us access to his stroke patients. So Rich, I'll close. We have work done with a variety of colleagues in neurosurgery and outside of neurosurgery who have helped with this. I'm more of the Pierre D or conductor. I can't do anything technical anymore, I swear. And I think in time, with a little more funding, we'll have a device to put in your hands that can help triage patients as they enter the ER, as they enter an ambulance, that can give a proxy for midline shift that can help with triage decisions. And Rich, that's the end of my talk. Thank you for this opportunity.
The brain is a 40-watt light bulb. And as most of you know, we've been part of maybe the past 2 decades of renewed interest in the neurosurgical neuroscience world and in what we can learn from human invasive EEG studies. And so that, when I started, was pretty much exclusively subdural arrays and various other subdural methods. That has evolved to include both that and the approach with the robotics, and just a cheap slide to get Dr. Buckley in a picture. But our ability or our use of invasive monitoring has only grown, and that this information that comes with these studies has tremendous richness of both strength of signal and also the ability to see very high frequency, low amplitude changes that are important for cognition. So I want to go over the basics of what we've done, which is now probably about 20 years old, and then some of the cool applications, and then wrap it up with a clinical application that we're doing right now. So folks, especially residents, are aware of the opportunities. So this is the general concept is that we have signals that are in this subdural or with stereo EEG intraparenchymal, but on the scale of somewhere around 200 to 500,000 neurons. So you're looking at very large populations. It's much closer to the action than a scalp EEG can be, signal not attenuated by skull soft tissue and distance, and obviously spatially sampling only where you sample, but tells us something that's intermediary between that and the single unit work that many have done with what's called BrainGate, or the microelectrode arrays that have been in maybe a dozen paralyzed patients across the country. So, what's the benefit clinically? You can see, as you see on the ICTL study, well before you could possibly see this on the scalp, you can see very high frequency changes that will often precede a seizure. And then you can also do a lot with the different EEG bands. So the terminology you start with a signal like at the top, and then you can break it down into its frequency components. The Greek lettering is historic, so a little bit non-intuitive, but we'll focus primarily on the beta rhythms, which is typically means somewhere in that 12 to 24 hertz range, and anything above it is generally called gamma. Those we'll talk about, that's probably capturing multiple phenomena, to use that terminology. So there's also a mu frequency, which is a form of beta over motor cortex. From the scalp, it actually is lower frequency, so down in about 12 hertz. And it's been known for a long time to be of interest because it's actually higher when you're at rest. And then as you do movement onset, it attenuates. And so that's what all these different power frequency curves are showing. I'll show a lot of these, but so I'm going to try to go over it each time. But you have amplitude on one axis and then the frequency on another. So somewhere in that kind of 20 hertz range, and it may bounce around in folks, you'll see a difference. And this is just a difference between movement and rest, showing you where which frequency captures that difference. And that's again been studied for a while. Huge industries, scientific industries around trying to model this and see what else is going on from the scalp. But it was really the Hopkins group in 1998 that nicely quantitated these phenomena that people have been seeing, and showed that whereas the in this case they looked at the sort of low beta, so they called it alpha, but whereas these low frequencies will drop with the onset of movement, if you look higher, maybe in this case they were the first to really quantitate going up around 80 to 90 hertz, then you can see an increase that correlates well with movement. And so what we did is put together a fairly large group, showed that if you're, as everyone knew, that if you look at these low frequencies and look at hand movement, which is the red curve, and rest, which is the blue curve, again, a power spectra that around 20 hertz, you see much more power in the signal at rest, and then that drops down when you start movement. But if you now, instead of sort of attenuating everything above the 60 hertz artifact, you open up your filters and look, which you can do with the increase in computational power that came in the mid 2000s, that allowed you to look at these very high frequencies. And what you saw is that both that there was this increase in power at these lower frequencies and or higher frequencies. And if you look at the amplitude plot, each of those hashtags or hashes of marks on the side are an order of magnitude. So this means that you're looking at things that are maybe 1,000th or even 1/10,000th of the amplitude of the high frequency. So when you look at an EEG or really any natural phenomena, typically the low frequencies dominate what your impression is, but hidden in that are these higher frequency signals. And then also what we found is there was no specific frequency there. So there wasn't one frequency that was associated with thumb movement or finger movement or other things. And that was one of the theories that was being kicked around at the time. So it looked like these high frequency changes were what came out of a very large population of many different timescales of different neuronal circuits and different interactions. And the other thing that was interesting is that the high frequencies were actually much more focal. So the low frequency changes would be over a network, say in motor cortex of when you moved your thumb, you might see the whole hand area and even mouth area have the low frequency drop out, but the high frequency was quite focal. And further, that when you talk about the gamma band, you actually were capturing both phenomena of this low frequency drop and high frequency increase. So we really started to talk about higher frequency signals, and then also focus on the low frequency as a broader, say, turning off, opening the gates for activity. And so, and this matched our understanding of cortical organization nicely, with the hand and tongue movement very separable by high and even by low frequency, but especially the high frequencies in a given person. And then another phenomena that I want to spend a couple minutes trying to explain, and I still struggle a little bit with the concept, so it's not super intuitive, but when you look at how these low frequency and high frequencies interact, you see, especially at rest, an interesting link. And this has been known from the primate physiology as well, is that when you look at low frequency cycles generally, so it could be beta, it could be alpha, it could be even lower frequency, the theta, which is 4 to 8 hertz. And then you at the same time look at how active a local cortical population is, they're linked, and they're linked by the phase of the lower frequency. And so when you, in general, when the lower frequency cycle is turning off, so this descending phase, here. So this is the waveform of the low frequency. So it's sometimes there's a lot of energy at that frequency, and sometimes there's lower. And as it's descending is when the higher frequencies seem to be more active. And so that's phase dependency. And it's typically in that phase, and you see a plot here. So depending on the phase of rhythm, and so you get this increased activity, and the degree to which that activity follows the cycle tells you about phase locking or phase encoding. And that relationship becomes interesting in some of the applications that we're going to do. So I'll come back to that in a second. What I first want to show you is what we did with some of these signals, because I started a collaboration around that time with the lab of Raj Rao in computer science and a variety of graduate students, starting with Kai Miller, who's now in neurosurgery in Mayo, and then a lot of other graduate students throughout mostly in industry throughout the area. And we started with this concept that these frequency changes, the high frequency changes, are very focal, and they can link them to a behavior. So you'd start with, say, hand or mouth, in this case, mouth motor cortex, and you say, okay, we know what the power spectrum is at rest. We know what it's going to look like when you're active, and we can find a frequency band where we can separate out the two signals based on those two states. So in this case, we picked this 16 hertz wide band. And then we do a very simple task where we have a cursor go across the screen, and we link the position of the cursor to the difference to the power in that electrode in that band. And so when the power is high, the cursor goes up, and when the power is low, the cursor goes down. And you give the subject a target, either up or down, and then they have to drive the cursor to that target in a set period of time. And there's lots and lots that we did with that, but the thing I want to focus on was the ability to volitionally control this, because we didn't know going in whether if you instead of thinking about the movement, actually thought about moving the cursor up or down, whether you'd be successful. And what we found is not only are you successful, but you can actually volitionally enhance the power in this area. So if you take an area like the example I showed you and look at the movement when you're actually moving, in this case, your mouth, and then even when you imagine moving your mouth, you have signal there, but it's weaker. But then when you do that feedback task or that cursor task I showed you, the power actually goes even higher, as you see over here in this bar, even higher than, see if I can get rid of the screen here, than when you were doing the overt movement. So enormous ability to modulate these cortical signals. So we have, again, we see this low frequency desynchronization, and these are and high frequency changes with the task. These two signals are linked by the phase of the lower frequency modulating the higher frequency independent of task. And we do see that volitional control is possible. So one of the questions, so I'm just going to spend about 5 minutes on a cool scientific question we asked. One of the questions is what goes on in cortical representation. Because cortical representation is if you look across species, is obviously very, the brain is dedicated to what's important for the organism and its environment. At least that's the interpretation, which is a reasonable one of what you see. So if you have whiskers, that's important. The other parts of your body that interact with the environment and for information. So your digits, your mouth for communication, and so forth, are all important. If you and that's why whisker barrels are sorry, I don't know if it's why, but you do see that whisker barrels are large in rodents, and that helps for information, and also why it's a good target for neuroscience. And then if you're a ground dweller, this is what your homunculus looks like, which I just like because it's kind of gross. But the salience is associated with the cortical representation, and we know that even in people that this can change. So it can change chronically. This is a model of study of amputees that using motor evoked responses through TMS, showing that in general that the face representation will take over or substitute in if there is no hand. And so we asked the question, is the homunculus static when we're doing these experiments? So what we did is a fairly simple task. We had a subject squeeze a the ball there to a specified force. So we knew we were doing the same task repeatedly. We then picked the electrode that had the strongest activation when doing this task. So we wanted to see what happens in the area that's most mapped to this task of squeezing the ball. And then we used that over about a 10-minute period to do the cursor task that I showed you, and then repeated the motor task. And wanted to look for changes in local activity. So what we saw is that, again, you have the activity, you find a sensory motor response that's that's strongest for that that simple motor task. Use that same signal to drive a cursor. And then when you came back, what you saw is that actually the activity in that area had gone down, and other parts of the brain were being used to perform this simple motor task. So that at least transiently, this piece of the brain was being, well, I'll say repurposed. We don't really know the mechanism, but that it was as if it was being used now to drive a cursor instead of use your hand. So it did appear that we did something with transiently with the cortical representation. So the next experiment we did was to actually try to see how flexible this process is. And so we did a concurrent cursor movement and motor execution task. So the way this works is you had these 8 targets. Up and down was driven by the cursor attached to motor imagery like we saw before. And then you used your hand to tap either the left arrow or right arrow. So using the contralateral hand, and that would move your cursor the different directions. So if you were trying to move it straight up, you wouldn't tap an arrow, and you just imagine moving the cursor up. And if you wanted to move the cursor to the left, you tap the left arrow and do nothing with the cortical control, and so forth. So center out movement of the cursor, and just what I said. But it's the same cortical area that drives both components. So we did arrow tapping, found the electrode that was most strongest for that, and then also had that drive motor cortex. So if you aren't able to dissociate cortical activity, then every time you tap the arrow, the cursor should go up. And there should be no way to drive the cursor down, which is no activity, and move it to the left or the right, because you'd be using that piece of the brain. So the only way that this would work is if you can completely dissociate the function from that piece of the brain. And so there's the design again. And here's what we found. So this is, if you look at the subjects, the important thing is that when you, so going up is fairly straightforward. That's just imagining the cursor. So it's all that red is the high activity, high gamma activity. We looked at that 70 to 90 hertz band that's between those 2 horizontal bars. If you look at the T1. And then if you look at T5, that's fairly straightforward too. That's low power in the brain. And again, you're just resting and you're not using your cursor. But here's the fascinating one. So both the left arrow and right arrow, you see baseline activity, even though you're now using that piece of the brain to move. And not only that, but you actually dropped out selectively this portion of the frequency. You see there's still some high frequency signal above the band that was used for control. And then perhaps even more remarkable are the ones where, again, you're moving the cursor and there's no activity. And here, of course, you're moving the cursor and having activity, so that's a little bit less exciting. So this is, again, you've completely dissociated the native function of the cortex, and you've done that in really just a matter of a few minutes. Okay, now, so our idea is that not only can you modulate, but you can actually add representation to your homunculus, and this seems to be a process, perhaps with any skill that you're learning. So, you know, we know that these high frequency changes associated with activity can be volitionally modulated. They may even be able to be eliminated. So what do you do with this? And there's a variety of applications, many of which we won't go into. You can spend hours on it. There's communication tools, therapeutic modulation, like we and others have done with like Parkinson. We had a trial with a Medtronic device where we used the beta signal to modulate thalamic stimulation. That was Andrew Coe and Jeff Herron. But then we're also going to use this and try to use this as a signal for cortical stimulation. So this is obviously where teams will come in. Here are my colleagues. I tried to find older, more embarrassing ones, but that can be used against you too. So But you know this crowd, Jeff Heron, who's our PhD and leader of the engineering efforts, and then Drs. Grant and Coe and Houtman in no particular order. Then the future, which is our PhD students, Courtney Pascal's MD PhD, Lila Levinson, who was a student in our their surgical summer program, and now a PhD in neuroscience. Samantha Sun, who's doing an internship at Microsoft, and then Monica Liu in the lower right, who is a Shannon scholar from Allen Institute, who's spending her time with us with computational work. And so what we decided to do was tackle this problem of how to improve the recovery after stroke. And this is data from a trial that's now probably 10, 15 years old. It was a local company, North Star Neuroscience, and they eventually got bought out and had different names. But what they did is did cortical epidural stimulation of motor and sensory motor cortex in those with stroke and motor deficits, chronic deficits. And what they found was a benefit, but the way the study was designed is that it didn't meet the primary endpoint, which was a shorter difference. And so what they found is that physical therapy versus physical therapy with stimulation, if you look at the upper panel, all groups, there's no difference at 4 or 12 weeks. There is a fall off at 24 weeks. That's not how the study was designed to look at. So this didn't meet the endpoints of the study. And then also interesting is if you break up the groups into those that have preserved motor response and those that don't, in other words, a marker of the severity of the stroke, those that still had intact motor response were able to maintain a benefit. That's the upper solid bar in the lower panel, whereas the ones that did not have intact motor tracks looked a lot like controls. And so we and many others believe that there's still potential for using cortical stimulation to enhance stroke recovery. But we also wanted to look at some of the science that was evolving behind that. And this comes from labs of Eb Fetz, who's been doing this work for over 55 years, officially retires next week. This was one of the studies he did about 10 years ago with Stavros Sanos, who's now out in New York. But they had 2 small subdural rays in a monkey, and they had done work previously showing that if you stimulate one neuron in response to the activity of another neuron, that the 2 start to have very similar receptive fields or response fields. So in other words, if it's a motor neuron, they start to fire in ways that reflect each other over time. And so they tried to do this with evoked responses and seeing if they could enhance connectivity between these areas. So slide's a little busy, but the concept's fairly simple in that you stimulate one piece of the brain, you get a response at a distant site. And if you repeatedly stimulate that first site, you enhance the connection between the 2. But that only seems to happen if you stimulate at a specific time. And specifically, if you stimulate at this trough of the low frequency cycle. So going back to this concept that activity is linked to the lower portion of the cycle. What they found is that if you stimulate it at this lower portion, which is panel A, then your evoked response, which starts out as the orange, becomes a much stronger response, so blue. So these evoked responses are somewhere in a few milliseconds after a stimulation, you see this measurement at a distant site. However, if they gave the stimulation at the peak of this low frequency, then the brain was not receptive to this activity and connectivity. And in fact, you got a suppression of the cortical response. And this was a pretty strong effect where at this is just another phase diagram, where at this depolarized phase, you got a conditioning effect, and at the hyperpolarizing phase, you did not. So we had a life science grant in 2008 and then a Keck Award, and tried to do this in people. And what we found in our subdural populations is that you had, you'd stimulate the green electrode, you'd see evoked responses at several sites, and then you would stimulate the green in response to activity at the red electrode. And specifically, we would try to stimulate at the depolarizing phase of the red electrode. And the idea was that that would enhance the connectivity. That's the black line with the cartoon synapse between the green and the red area. So the area that you're stimulated will link to wherever you're recording from as you're stimulating based on the phase of that signal. And this is what we found. This is what the tracings look like. You see the evoked response peak, and then the red is with the conditioning effect that we show. That may not seem like a lot, but it's wildly statistically significant. And so and it was a dose effect. So if you did 3 stimuli in a row, then you got this 3 microvolt enhancement of the evoked potential magnitude. So this was to us evidence that we were able to get pieces of the brain to connect more. Could you do it in patients who had motor deficits? So we had the opportunity to look at a patient who had not great hand function, but had some wrist flexion and shoulder movement that he wanted to preserve. So he was not interested in a radical unmapped approach. And we still had some questions about the localization of his epilepsy. So he had electrodes implanted. First, we found that perilesional areas had the same profile of activity with movement. So this high frequency increase, low frequency decrease. And second, we found that we could find the same effect with stimulation where we could enhance connectivity between areas. So this evoked potential enhanced in the setting of prior stroke. And then over about 7 subjects, we looked and found what we believe to be a similar effect. It meets statistical significance, although you can see it's not quite as crisp as the non-human primate work. But this is that when we delivered the stimulation in the hyperpolarized state, we did not get an enhancement of evoked responses, whereas we did when we were in this depolarizing state. So this phase dependency, again, on the beta rhythm did seem to drive this effect. So our goal, and we recently, after 3 rounds, got a Brain Initiative grant to do this, our initial goal was to try this with a new device that comes out of Germany, where we would implant and then stimulate during rehabilitation, similar to the North Star trial, but do it using this closed loop stimulation with the idea that we would be bringing in areas. We don't have any further animal data suggesting this. And after our first round with the FDA, they thought we were stretching too much with this first. So we're going to start with this protocol where we do the rehabilitation, try to mimic the North Star trial, prove that we can do closed loop system work but without therapy within the follow-up. If we pick our patients right, there's still benefit to having this implant, but then would allow us to do show that this closed loop system also is effective, and then eventually compared directly with larger trial. So this will be exciting because in addition to the neuroscience behind it, it's very likely, we know that folks can modulate these rhythms. So if there is an adaptive effect and they understand that, they may be able to enhance it on their own volitionally and quickly. So that should keep us busy for a while, assuming we can get through the FDA process. There have been many PhD students, postdocs, and then I just highlighted a few of those with neurosurgical connections. A young Dr. Buckley, Dr. Collins, who's faculty at OHSU and had a K-12 award to study microelectrodes in awake patients. David Su, and then Hai Sun, who's now interim department head at Rutgers in neurosurgery as well. So